a great pleasure to have Ray Mansbach here today for our second in-person colloquium this year. Oops, sorry, I lost my note. So Ray did their PhD at University of Illinois at Urbana-Champaign until August 2020?
No, that's
wrong.
2018.
Yeah, 2018. Okay,
yeah.
So minus one. Then moved to Los Alamos National Laboratory for 2 years as a postdoctoral research fellow. And Ray has been assistant professor in physics at Concordia University, Canadian Research Chair in computational biophysics since August 2020. And as you can see, that was not the easiest time to start a position, I think. Ray has been doing wonderful work at the intersection of 3 domains which are very close to my heart, I have to say. I think you can see that here. It's both first statistical mechanics, and then biophysics, and then machine learning. And I don't know what Ray exactly what will tell us about today, but I hope that this will convince you that those fields are really, really closely interconnected, that there are many interesting connections. And so we're very happy to have Ray with us today as a Montrealer as well, and building connections between our universities. So let's welcome Ray.
If you feel comfortable, as long as you're 2 meters away, you can take off your mask when you talk, but it's up to you.
All right.
No pressure.
I don't know, I might just leave it on because I'm neurotic. Thanks for that great intro. Forgive me if this talk is a little stumbly. I have not given an in-person talk in a year and a half or so. So it's a bit of a first. But what I'm going to be talking about today is what I've sort of fancifully called learning from life. I'm going to discuss my motivation for getting into the field that I'm in, broadly speaking, and touch briefly on some of the tools that I use. Then I'm going to talk about 2 projects I worked on. There's actually going to, it's going to turn out to be 3 projects and a half, but 2 projects that I worked on during my postdoc. That's going to be what I spend most of my time on. And then I'll say just a little bit about where I'm hopefully going as my lab attempts to get started. And then I'll conclude. So when I was 13 years old, I read The Lost World by Michael Crichton, which is the sequel to Jurassic Park, and just like Jurassic Park, it's full of lots of big stompy dinosaurs. And I was terrified by The Lost World when I was 13 years old. And now I'm going to ask the audience a question. Given that the hydrogen atom is on the order of 1 angstrom, how big do you think the thing in The Lost World was that little me found the scariest? So everybody who thinks 10 angstroms, raise your hand. Okay, 1 for 10 angstroms. Who thinks 100 angstroms? Raise your hand. Okay, we've got some people around here. 10,000 angstroms? Okay, people are leaning towards the big 10 to the 11th angstroms. A lot of people voting for 10 to the 11th angstroms. Well, you're wrong. In fact, it was the 100 angstroms people. But here are the things that are on these characteristic length scales in The Lost World. And the reason I'm talking about this is because I'm going to get into talking about things on these characteristic length scales during the talk. So good old Tyrannosaurus rex is, of course, 10 to the 11th angstroms, and she is very scary. But she is not as scary as the prion protein, at least not to me. So there's also morphine, which is a small drug-like molecule on the order of 10 angstroms, which is given to one of the scientists as a painkiller when he falls and breaks his leg. And then we have the prion protein. So I'm assuming we have a lot of people in here who have a pretty solid biophysics background, but in case you don't, the proteins are little molecular machines in your cells, and a lot of them act because they fold up into a particular shape, and that shape is responsible for how they can interact with other proteins. The prion protein, like some other proteins, can also fold into what they call a rogue conformer. It can misfold, and when it misfolds, it will assemble into these long fibrils that are on the order of 10,000 angstroms or so. And this will template more misfolded proteins. And that will cause neurodegenerative diseases, which in fact kill the dinosaurs, which is why I found them terrifying, because I was like, how can something so tiny have such an effect on something that's so huge? And so as I went on and researched, I ended up looking at small things that kill big things, although not really small things that kill things as big as, well, some small things that kill things our size. Most things the size of T-Rex aren't really around anymore. But this is kind of the 3 length scales that I look at currently, or that I have been looking at. I still have a project on small molecules. I'm going to talk to you actually about how I did a little bit of antibiotic design of small molecules to kill gram-negative bacteria. And then my first love is still proteins. I love proteins. I think they're super cool. And those tend to be still on the order of 100 angstroms, but the ones I'm looking at are, the ones I'm going to talk to you about today are toxins. So those still kill big things, us. Although I want to use them for therapeutics. And also antimicrobial peptides, which I won't say as much about, but which is what is actually one of the big focuses of my lab as we try to get started. And antimicrobial peptides also assemble like prion proteins, but in this case they do something very nice for us because they are created by the innate immune system. And what they do is they form large assemblies on the cell membranes of bacteria, on the order of 10,000 angstroms, roughly, puncture them open and kill those cells. So you can see a lot of, currently I just kind of ended up looking at a lot of things that are potentially antibacterial because I think they're really interesting. So the way I do this is I use molecular dynamics, which I like to call, and I've seen many other people call it an in silico microscope, because essentially what you're doing is you're solving Newton's laws of motion for a bunch of molecules and to understand how the proteins, for example, might fold or not fold, or how you could design those things to fold. And then the other thing that I'm interested in using, the other method, is machine learning, which I quite liked this definition that I got from the MIT open courseware, where they said machine learning is a subset of artificial intelligence. Artificial intelligence is the ability to process information to inform future predictions. Machine learning, which I will talk a little bit about in this talk, is the ability to learn without explicitly being programmed. And there are very, very simple ways of learning without being explicitly programmed. In fact, I'm basically just going to be frankly talking about what I did at LANL was mostly just linear regression. But it's still machine learning. And I'm hoping to get, as I start my lab, go into some stuff that's maybe a little bit less simple, which is deep learning, where you use neural networks to extract patterns from big data. Again, without explicitly being programmed, but I would like to, because I'm a physicist, really get a better understanding of how deep learning works, although I won't have a lot of time to say much about that in this particular talk. So overall, my overarching research interests are bringing together physical model predictions with machine learning tools to perform physics-based design for complex biophysical problems. Now, having said that, the first project I'm going to be talking about focused a little bit more on the machine learning side. The second project focused a little bit more on molecular dynamics and physical model predictions. My lab, as we start going forward, we're going to try to bring those together. Again, I haven't had as much of a chance to get started, perhaps, as I would like, because, well, starting in the middle of a pandemic, I'm traumatized, my students are traumatized, we're working very slowly, but that's okay. All right, so gram-negative bacteria. These are responsible for some of the most intractable bacterial infections due to the combined effects of their highly impermeable outer membranes, which make it really hard for drug molecules, for example, to get into the cell to kill them, and the efflux pumps that will actively pump out molecules that do manage to get inside that outer membrane. So when people go try to design antibiotics that kill gram-negative bacteria, you end up having a lot of molecules that fail. It's really slow. It's a leaky pipeline, and there's a lot of miscellaneous information on molecules that failed in one way or another. And so when I got to Los Alamos, the problem that I started thinking about was how do we rationally design drug molecules to bypass the outer membrane and target the cells? And my idea was, well, can we find a vocabulary of antibiotic action? In other words, can we find parts of a molecule that are responsible just for being able to get into the cell, and parts that are responsible for it just being able to stay inside the cell, and then potentially parts that are responsible for it actually being able to kill the bacteria. And if you can find those fragments, I thought, well, maybe you can make that into a kind of vocabulary and sort of recombine them for hybrid antibiotics. And the thought was, well, then we can also reuse data because we might have some molecules that have aspects that we want, even though they didn't end up being good at killing the bacteria overall. So I ended up doing mostly a proof of concept on this, and we focused primarily on just permeability while I was there, although there's been a little bit more work, and I have done a little bit more work as well since I've been gone. I won't be talking about that today. What I'm talking about today is the sort of early proof of concept hunting fox for hunting fragments of X analysis. It's always very important to have a cute acronym for your algorithms because then people like them more. And so here is basically how it works. We have a series of drug molecules, and our experimental collaborators measure some drug activity. In this case, a drug activity related to how well they permeated into the cell. And then we cut the drug molecules up into fragments that make them up and perform some fairly straightforward sparsification and hierarchical cleansing to do feature selection in a traditional machine learning-y kind of way to identify active submolecular fragments, which hopefully in the future will inform drug hybrids. And also, we took these active submolecular fragments, or fragments that contain information about how well the molecules permeate, and trained another simple machine learning model to be able to predict whether drugs can permeate. So the idea is now, okay, we can use this model to predict on drugs that haven't been experimentally measured whether they're good at permeating into the cell, and that's really great because essentially it's a way of narrowing the search space in an initial phase for the experimentalists, which is always something that I think is really important. So I'm always really big on both search spaces and the power of computational methods to be able to narrow those search spaces so that you're not searching through a million, maybe you're only searching through 500,000 molecules, right? Which is a lot easier to do, still hard, but. So here's our fragment-based representation. It is the most naive thing that I could think of because I'm a physicist and not a biochemist. I learned a lot during my postdoc, and now I know a little bit more biochemistry, and this is probably not how I would do this now. This is how I did this then. Basically, I just took a sliding window, went down the molecule, and chopped up the molecule into all the fragments that made it up of various different sizes. So what I'm showing you here are two example fragments, one of radius 1, one of radius 2. I went all the way from radius 1 up to radius 5. And so then I made a fragment-based representation. So I did that sliding window down all of the molecules that we had experimental data on. I chopped them all up into all the fragments that made them up. I threw those fragments into a pot, and then I represented every single one of those molecules in terms of the frequency of times that a particular fragment showed up in the molecule, which might be 0. So this ends up being a sparse, naive representation, a 22,000 length frequency vector for each molecule, and a lot of those frequencies are in fact 0. But it is a representation, and it is fragment-based, which is what I wanted it to be. So then, how did the experimental collaborators measure molecules that were good at permeating the cell? Well, what they did was they made these two mutants. So we have this wild type gram-negative bacteria that I said has an outer membrane that has these efflux pumps. And they actually created two mutants, one called P delta 6, where they knocked out the major genes to get rid of the efflux pumps, and one called P delta 6 poor, where they did that and they also hyperporinated the outer membrane. And the nice thing is, if you do this, you can now measure drug activity on both of these mutants. The way you do that is you measure what's called the minimum inhibitory concentration of a drug, or the MIC value. And the MIC is just the minimum amount of drug you need to inhibit the growth of the bacteria. So the lower it is, the more effective the drug is. And then if you look at the ratio of these things in these two mutants, if the outer membrane has no effect on the drug activity, this ratio should be 1, whether or not it's good at killing the bacteria, because it's getting inside and it's acting the same way whether or not the outer membrane is there. If the outer membrane is seriously impeding the drug, then this should go to basically 0, because this has a low MIC, and this doesn't have a low MIC because the drug is stopping, the outer membrane is stopping the drug from getting into the cell. So that's our, let me just go back quickly so I can. So that is our fragment-based representation and our drug activity. The next thing we have to do is feature selection. Like I said, these are 22,000 length frequency vectors. And so they're very long, and a bunch of these fragment frequencies are zeros or irrelevant, or don't have the right information, or they're fragments of inside other fragments. So we basically had to perform a machine learning technique, and I don't have time to go into great detail, but called sparsification. Specifically for people who are machine learning-y, we just used lasso regression with an L1 penalty, and then we used something to remove hierarchies between the fragments. So then what we had was we had a function that takes in a molecule as a fragment frequency, but now of the fragment frequencies that we say, these are probably important, that outputs a prediction for that ratio, or for whether essentially this thing is going to be good at permeating, or whether it's not going to be good at permeating. And that's basically the full algorithm. We ran it 28 times for model voting to see how robust it is on different slightly randomized training sets, so basically bootstrap analysis. And at each step we identified a set of non-hierarchically related fragments, and we identified a set of expected to be highly permeating molecules from an external library. And the first thing we did was we just tested, and again this is proof of concept, to make sure that the model does work, which it does. So what you're looking at on the top there is what's called the receiver operating characteristic curve. Essentially, this is a measure of how good a classifier is. It's the true positive rate versus the false positive rate. So if that classifier is an all-seeing oracle, it always gets everything right. It has 0 false positives. It jumps immediately to true positives, so you get a step function. If the classifier is just guessing, it's not an oracle at all, it doesn't know anything, then the true positive rate is equal to the false positive rate, and you follow along that line of randomness. So the closer you are to a step function, the better. In this case, our model works pretty well for things that don't permeate. It still works for things that do permeate, not as well, and unfortunately the reason for that is, well, we had a lot more examples of things that were good at, we had a lot more examples of things that could not get into the cell than things that could, but it was still doing a pretty decent job, and this is important, this is the enrichment. The enrichment is the measure of how many more hits our classifier finds than we would find by searching randomly. In essence, it's a measure of how much we've narrowed the search space, which I said is something we care a lot about. So an enrichment of about 100%, we have over 100%, means you find twice as many hits as you would if you just searched randomly through the search space. In other words, that corresponds to halving the search space, which is pretty decent. Not as good as it could be, definitely something to keep working on, but pretty good. We also did actually pull out those fragments. So what you're looking at here is a histogram of the number of fragments that showed up, a number of times, how many times they showed up. So like, for example, here, we had one fragment that showed up in 25 out of 28 of the bootstrapped runs, and then we had, unfortunately, a bunch that showed up in only 1 or 2. And the idea here is, okay, we're not as robust as we might like to be. You would like to see more fragments showing up more often and not be quite so dependent on the training data. But we are seeing things showing up repeatedly, and the things that were showing up repeatedly do have clear interpretations biochemically, despite the fact that we didn't include any expert knowledge beforehand. I've noted a few down here, but the one we ended up going and looking at a little bit, the aromatic groups and the amines, I don't have time to talk too much about this. We did do some molecular dynamic simulations to see that part of the reason that this makes sense is, for example, the amines, they're charged, tend to be positively charged. Bacterial cell membrane tends to be negatively charged, unlike a eukaryotic membrane. It's negatively charged, so molecules that had fragments with higher amine counts tend to partition more tightly against the bacterial membrane, so they're stuck on there basically. So they're likely to permeate just because they're spending more time there. We also, like I said, predicted some possible molecules that had not been experimentally measured. And this was great because we were actually able to experimentally verify that our algorithm is doing what we wanted it to do. We pulled out 7 hits that showed up in more than half of the iterations from 31,000 external molecules, tested them, and 4 of them were, in fact, permeators as we expected. 2 of them were in the library, but for some reason my experimental collaborators actually couldn't get a hold of them. I think there was like a supply line issue or something. And then one of them was actually unmeasurable. So this was a slight problem in the assay where you measure the minimum inhibitory concentrations in the two mutants. But if the thing doesn't kill the bacteria at all, you just get a nan over a nan and you can't measure anything. And so we think this probably does permeate, but this actually led us to say, okay, we also need to do some other experimental assays to actually be able to tell if they can get in without them having to have some activity against the bacteria. And we did actually do that. There's a second paper on something related to that, but again, I'm not going to spend too much time on that today. But overall, what we were able to do here was here's my hunting fox algorithm. This is why you always need to have a cute name. Diving into the snow and pulling out fragments for fragment-based drug design. So we validated a broadly applicable algorithm for drug design and data repurposing. We understood the fragment-based side of permeation. And the hope is that in future we can do some de novo design of antibiotic hybrids from relevant fragments. Now I'm going to totally shift gears on you and talk about protein folding and design, which again, as I've mentioned, is my first love. And this is where we're going to have a bit of a surprise partway through. So, venoms. Venoms are really cool. This is actually another thing that comes up in Jurassic Park. Well, The Lost World, actually. So conotoxins are venoms produced by cone snails that help them, that basically paralyze their prey. And oh, the reason I have a picture here of a knight fighting a snail is because the knight is me. I'm trying to wrest the secrets away from the snail. But really the reason that this is here is because this is actually kind of a medieval meme. There's just knights fighting snails along a whole bunch of medieval manuscripts. Nobody knows why, but I was just like, it's really cool. It's a knight fighting a snail. So that's your trivia for today. So conotoxins are a good source of therapeutics. They are small and they're disulfide-rich. So basically they have a bunch of cysteines and the cysteines get connected by covalent disulfide bonds. And these kind of staple the conformations together, so they're small, very stable, and have very specific conformations. And that gives them a very high selectivity for ion channel receptors, which makes them attractive targets for biomedical design. In fact, there is currently a novel analgesic on the market called ziconotide that was derived from the venom of the Conus magus snail. And so what I wanted to do, or what I still want to do actually, because this is kind of ongoing work in my lab at this point, is to be able to develop the class of conotoxins as scaffolds instead of just picking one and going along with that. Again, I'm going to start talking about mapping out search spaces. We need better unified predictions and better understanding of small disulfide-rich peptides as a whole. Because I said they have these disulfides that sort of staple them together. And there are multiple ways to connect these disulfides. So for example, if you have 4 disulfides, you could connect the first one and the third one and the second one and the fourth one, the first one and the fourth one, the second one and the third one, the first one and the second one and the third one and the fourth one. And if you have 6, you have a lot more connections. If you have 8, you have a lot more connections. And the thing is that different connectivities with the same sequence have different structures that act in different ways because they're different structures. And in fact, different sequences that have the same connectivity have pretty similar structures, like you can see on the bottom here. And it's not entirely clear whether these things have a single native conformation or whether they essentially populate a metastable ensemble of multiple conformations. And so that's something that I was interested in. And there was a recent-ish, 2018 I think, study on the mu conotoxins, which are conotoxins that block the sodium channel receptors, where they tried to rank them in terms of whether they folded in an ordered or a disordered way. They were able to kind of link them. The paper kind of showed that the ones that folded in a way that was more ordered tended to have more of a single native state. The ones that folded in a way that was more disordered tended to have kind of a metastable, like you had a bunch of structures, different connectivities, showing up in solution, essentially, because either they were all roughly stable or they were kinetically trapped, like they were stuck that way, basically, on long enough timescales. So they call the more ordered ones BPTI-like, because BPTI is a small disulfide-rich peptide that folds in a very ordered way. It just kind of says, okay, my first native disulfide clicks together, folds, done. Second native disulfide clicks together, folds, done. Third native disulfide clicks together, folds, done. These are the ordered ones. Then the more disordered ones, they say, fold more like pyridine, which is just like, I'm going to collapse because I'm in water, boom. And then it connects all the disulfides in some random order, and then it reshuffles them. But reshuffling disulfides means you have to break and reform covalent bonds, so you're much likelier to get things that are either kinetically trapped or actually metastable. And so I kind of said, okay, can we actually compare these in sort of a unified free energy landscape way? And the way I did this was by using something called the diffusion map, which is a nonlinear dimensionality reduction machine learning technique. That's a lot of words, but I'll explain it in kind of a high-level idea quite quickly now. So the idea is if you have some peptide, and this unfortunately is not a video of a clonotoxin because all my videos of clonotoxins are stuck on my computer at Lanel and I can't get them off for a variety of reasons involving paperwork. This is an antimicrobial peptide. But anyways, you can describe these configurations as points lying in a 3n-dimensional space because you have n atoms, they have 3 dimensions, okay. However, you don't really need 3n variables to describe these things. You could just kind of say, well, it's in a helix and the helical angle is this way, and that mostly tells you what that conformation looks like. In other words, in a slightly more deep way, you can use or rigorous way, you can use the Morris-Wanzig formalism to show that most proteins fall into 2 to 5 dimensional nonlinear manifolds. Or in other words, you can use about 2 to 5 variables to describe the conformations of most folded proteins. The idea is that these are proteins, these are points in a higher dimensional space that lie on a lower dimensional manifold. So this is an example, the Swiss roll, it's not actually a protein, but this is the idea. You've got these points that are in a 3D space, but you don't need 3 variables to describe them. You can just describe how far along that manifold or how far across that manifold they are. And the way you can pull that manifold out is by using this thing called the diffusion map, where you essentially mathematically simulate a diffusion process across the data. So you need to know some kind of dynamical proximity of the points. How easy is it to transition from one point to the other? And if you have that, just like applying ink to this bird, just as that sort of diffuses across to reveal the structure of the bird beneath, if you apply the diffusion map to this data, it will pull out the hidden structure of the data beneath it. And so that's what I did. I took these 5 conotoxins, I disconnected their disulfides because I was interested to see how much the sequence would dictate the underlying folding landscape and whether that would tell whether there was a way to kind of map that to ordered versus disordered. And I ran replica exchange molecular dynamics simulations. Then I did this little sequence alignment so that I could define some kind of distance between the configurations of even sequences that were different. So basically the distance metric for how different are these things was, okay, you take configurations of 2 conotoxins, you translation rotationally align them, and then you look at the root mean squared deviation of things that match in those proteins, basically. And the things that match that I defined are the heavy atoms and the 4 cysteines, because I lined up the 4 cysteines because that's important, and then the backbone carbon alpha atoms of the other things that match in the sense of not actually being a gap in the sequence. And what that lets me do is it pulled out a 2-dimensional description of all 5 of these conotoxins. And here I am visualizing the first variable that is most important in describing their folding. Blue is basically one end, red is the other end. So you can kind of see this is, okay, extended on the one hand to little ball collapsed on the other hand. It's basically how extended or collapsed is the protein. The second important variable is this kind of, am I hooked like this or am I hooked like this? it's kind of it's a hook and slide mechanism. I've actually seen this before. So these two collective motions are really similar to the collective motions you pull out if you look at just like simple biomolecules like alkanes with a diffusion map. This basically is it's kind of a hallmark of hydrophobic collapse. And so with this, I was able to build this free energy surface where it's basically in the same basis for all 5 of these conotoxins, and then look at the differences between the free energy surfaces. They're low dimensional, like I said, they were low dimensional. They're flat, which suggests they're fairly disordered, which kind of makes sense because they don't have the stabilizing disulfides clicked together in them. And they're pretty similar, but there are some different details. So what I've marked on these free energy surfaces, this X, is the approximate location of where the quote-unquote native conformation is projected into. So the one that you found with the people found doing NMR structures that they defined as the single native state. And what you can see is that for, particularly for the 2 most disordered, the minimum of the free energy landscape is pretty close to that native state. Whereas for the ones that are more ordered, the minimum moves away, particularly for the most ordered one, the minimum moves away from the final, where the final native state would be. And this makes sense because for that, what that's telling you is essentially the ones that are most disordered really are just collapsing down right away, and then they don't connect their disulfides because we're doing traditional molecular dynamics, so we can't simulate chemical reactions, but they're basically very close to the native state because they're collapsed into a ball. But the other ones, the ones that are more ordered, are just kind of hanging out over here, waiting to connect their first disulfide, which they never do. So this makes a lot of sense, and we were just about to start getting into doing some more in-depth analysis, and looking at like secondary structural propensities and things, and then this happened. So I'm about to shift gears again, and this is why. But just briefly, what I did here was used a unified dimensionality reduction technique to compare these equilibrium pre-folding surfaces in the muconotoxins to identify potential folding pathway predictors. And we did see that there's a potential predictor in how far along, basically where the free energy landscape is compared to how collapsed the thing is. And the differences we see also align with the expected disordered propensities, and it's also consistent with the hydrophobic collapse of pyridine-like peptides. So we're still working on this. It's gone a lot slower because I'm here now, but this is still being worked on. Sort of finishing up and finalizing the results in a little bit more of a fully finalized story-like way. But yeah, at this point, COVID happened, and all of a sudden I was not finishing up my work on conotoxins like I'd expected to be. I was doing work on COVID because so at Lannel, I worked down the hall from Betty Korber, who is a fairly well-known immunologist. She does work on, or she did work on HIV vaccines. Her team saw this mutation happening right at the beginning, and we're already worried about it, the D614g mutation. And it immediately started to take off. We started getting reports that the G form, there's a single amino acid shift from an aspartic acid to a glycine in the spike protein. We started getting reports that the G form was more infective and transmissible than the original D form. And that's when I got roped in because I'm a molecular dynamics person, among other things. And so I said, okay, let's try to figure out what the underlying molecular level underpinnings of that. How do we get from a single amino acid shift to a massive difference in infectivity, which is what people were seeing. And so I did just vanilla large-scale simulations of the full spike trimer. So the spike trimer is the part of the virus that gives it the characteristic corona, and it's responsible for viral binding to the ACE2 receptor. So the receptor binding domain of the spike protein interacts with the ACE2 receptor, and when that happens, this S1 part of the protein comes off the top and releases this spike that pokes into the cell, and that allows for infection. And it is a trimeric protein. It's made up of 3 identical subunits called protomers, and experimentally we knew that they can basically have one of 2 states. They can either be down or they can be up, each one. And we also knew from experiment that generally only one was ever found up at a time. You didn't usually get 2 pointing up or 3 pointing up. So what I did was I did 5 replicas of molecular dynamic simulations of the D form in the all down and the one up form, with one of the promers pointing up, and the G form in the all down and the one up forms. And so the first thing we saw was something we kind of expected, which was that the all down state is not what you want to be in if you want to bind the ACE2 receptor at all. So if you look at looking down from the top, this yellow part is the binding site region, and this, when we're in all down, it's basically occluded by these sugars that cover the top of the protein, and when we're in one up, it's not occluded anymore, so it can fruitfully bind with the ACE2 receptor. We didn't really see a difference here between the D form and the G form, so you're saying, okay, this is not responsible, there's not like a major change in, there was a little bit of a change, but there wasn't like a major change in the receptor in the exposure of the binding site from the D form to the G form. We didn't really see a difference here. Where we did see a difference was when I looked at the pattern of interprotomer contacts. So what you're looking at here is the number of contacts between protomers of the S1 and the S2 domain. So that is basically along this line near the mutation site here. And what you can see is that for the G form, when it goes from all down to one up, there is not like a major change in the numbers of contacts. There's a little bit of a change. Maybe you see some slight shift down or some slight change. You don't really see a huge change. They're all basically within error bars of each other in both cases. They're fairly symmetric. In the D form, though, you do see a change. It starts out symmetric in the all-down state, and then you go to the one-up state, and now you have a pretty strong asymmetry. And this corresponds to the fact that when it's an aspartic acid, when you have an aspartic acid at 614, as in the D form, it can form a hydrogen bond with 3 and 859 on the other protomer. And we see that the hydrogen bond occupancy changes, and it's asymmetric, in fact. This is this green thing, which you can't see very well, but it's there. So there's a difference between the all down and the one up, and there's an asymmetry in the occupancy in the one up state. But the glycine can't form a hydrogen bond. So it's just symmetric because it's 0 and then it's 0 again. So probably the reason that there's this change in the interprotomer contacts is because there's this loss of this single hydrogen bond that people have thought about possibly. I don't want to say too much because it's kind of a hot area of debate, but people have talked about it as maybe kind of a hook or like straining the protein a little bit in a sense. But anyways, this is probably responsible, and it results in this change in the numbers of this asymmetry in the numbers of interprotomer contacts, which results in a difference in the interaction energies, because a lot of interaction energies are just from how much you're touching, basically. I mean, I'm simplifying it, but we did check, and it does. We do see like an asymmetry in the D form interaction energies and a symmetry in the G form interaction energies in the one-up state. And this is the point when I got really physicsy and was like, well, look, these things can point up or down. There's 3 of them, and they interact. So let's call it an Ising model, because the whole world is an Ising model, because my committee chair was Nigel Goldenfeld, and if you know Nigel Goldenfeld, you know that the whole world is an Ising model. So, okay, Ising model, what does that tell us? Well, it can give us the ratio of the populations in the one-up to the all-down states. It's a 3-spin Ising model, so it's the simplest stat-mech problem in the world. You basically just say, well, there's 3 ways to get one up, there's 1 way to get all down, and then we have interaction energies that change depending on whether you're down, down, or up, up. Or up, down, or down, up, right, okay. And you can subsume these energies into just a single delta E, and we knew experimentally that in the D form, we saw a ratio of 50-50, the population ratio of 50-50. And I said, okay, well, we also see this symmetrization in the number of contacts, so maybe that means that delta E, which you get an actual delta E, you can just go through and you get some number for it if you have a 50-50 plugged in there. I said, well, we've symmetrized the DD, the up-downs now, probably. That probably means delta E goes to 0, which would mean that we would have a population ratio now of 75 to 25, and we just said, well, 1 up is the infection-capable state, So we have a thermodynamic reason that getting rid of that hydrogen bond leads to these changes in these interaction energies, and then thermodynamically you just have more things that are infection capable, essentially. And I said, well, that's a nice theory. So we went to our experimental colleagues and we said, we think a lot of the reason that this infectivity is happening is because there's going to be a shift from 50-50 population to, oh, say about 75-25. Is that what you guys see? And they said, no, 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 it's not 75-25, it's 82-18. At which point I had to take a moment because I have never in my life had a result that came that close. And it was the silliest model I could think of, right? So, okay, this was very exciting and very cool in the middle of a lot of stuff that was very stressful. But basically what we showed was that this enhanced infectivity is at least partially, there's probably some other effects of like kinetic trapping as well again, but at least partially due to just this simple thermodynamic shift that leads to a heightened population in the binding potential. So that was very cool. So that's my postdoc in a nutshell. How does that map to what I'm trying to do now? Well, like I said, I did machine learning on antibiotics. I did physical model predictions on chronotoxins and SARS-CoV-2. And now I'm trying to bring those together to do some physics-based design for complex biophysical problems. So specifically, still interested in bacteria. There's been a lot of recent cool work on using deep learning for drug design, for small molecule drug design. So the idea at a very high level is we have deep learning things that can create images that look like people that might fool you into thinking that they're real. What I would like to be able to do is train a deep learning model that is smart enough to create small molecules that trick, quote unquote, a bacterium into thinking that it's dead. So, anyways, there's been a lot. This has been a really fascinating area. In about I think this paper's from 2018. Yeah, so this came out in 2018. This is from Ellen Espiro-Guzik's group at Toronto, and they did this really cool deep learning thing where you can just get a deep learning model to come up with basically a low-dimensional search space, and you can move through that search space and generate new drug molecules. And I'd like to apply this to antibiotic design, so I'd like to kind of take that fragment-based idea that I was playing with during my postdoc and bring in the deep learning side so that I can also come up with some kind of latent space for small molecule antibiotics and maybe be able to predict fragment properties and actually see how you put those fragments together to make new antibiotic hybrids. We're not really very far on this yet because the one student I have working on it just arrived from India 2 days ago. But this is the idea. I'm also really interested, like I said, in antimicrobial peptide design. We're getting a little further on this, but we're still not very far. So there's been a lot of work on this too recently. I actually did a little bit of work on it in my PhD, again with the diffusion map in that case. And what I'd like to do here, again, this is kind of high level and we're not very far yet. We're still mostly up in here and just trying to get this part working. But basically the idea is, again, find a good deep learning model to generate peptides, find an interpretable, hopefully low-dimensional search space that goes with that deep learning model so you can push yourself around in, and help with that interpreting of that space by coupling it to multi-resolution molecular dynamics in sort of an iterative way. So ongoing work, still not very far. Also, I have a student still looking at the chronotoxins and trying to do something similar, maybe trying to do some landscape engineering to try to push those metastable ensembles to one way or another. Again, that's kind of pie in the sky, but that's where we'd like to go. And finally, in case, just so you know that I also do things that are completely off the wall, I'm looking at, I have an experimental collaborator at Concordia, Claudine Gauthier, who does medical imaging of brain vasculature. And I'm looking at, again, this is basically proposal stage, but I think it's really cool, so I'm telling everybody. Basically coming up with network-based simulations and using some topological physics models to try to predict the experimental brain vasculature maps with the idea of trying to figure out the way that COVID actually enters the brain. So I'm hiring, probably. I have 1 to 2 openings coming year for somebody with a solid background in statistical physics or computer science, biochemistry knowledge, big plus. This lab poster, this is my lab poster. it is something that I think is very important, so I'm throwing it up here. But if you're interested, come talk to me. I love talking to people about my research. The lab poster comes from, it's a Creative Commons. Creative Commons comes from this website, sammycata.com, which is basically there to show that racial justice and support for marginalized communities cannot be separated from the practice of science, which I think is very, very important. So overall, what have I talked to you about? Probably too many things. Multiscale modeling and machine learning to learn from life, I hope you think now is maybe a powerful way of thinking about understanding and design of complex biophysical systems. I've talked about how I might approach it in the future, how I've done bits and pieces of it in the past, and where I'd like to go with it going forward. So at this point, I would like to thank funding various collaborators and my current lab group who are all amazing. And I'd like to thank all of you for listening, and I would be happy to take any questions.
Yeah, okay, so I'm going to take care of the questions part, I guess. So is there any question? Lucas.
So you mentioned that you had like a radius for when you were searching for the size of the motifs. Do you expect to increase that radius? You might be able to detect more motifs that appear in like larger fractions of the bootstrap?
I mean,
because it seems to me
at some point, if you increase the radius enough, you just get the whole molecule, right?
Well, yeah, but also it seems to me that at least that one motif that you showed is quite small.
Yeah.
And I think you need something kind of larger to.
I mean, to be honest, like I said, this is pretty naive. What I would probably do now is actually search for pharmacophores, search for. Some people have actually done some nice work on identifying active fragments, and I've talked to a couple people about that. That's probably the way I would go with that now, as opposed to doing that really naive thing where you just chop it up into everything. But it's possible.
May I ask a follow-up question? Because so of course you look at motifs, but you could also look at contiguous motif in the molecule, for instance. In Shannon's book, there's a very famous experiment where you look at layers and doublet, triplet, and so on and so forth. So is it something that that you could consider? And I guess in terms of chemistry, you would expect that the actual sequence, the order will matter.
Yeah.
And you completely get rid of the order here.
Yeah.
So to some extent. So. Can you come? I mean, it sounds like very impressive that it works already like this. But do you think it will matter to look at the order, or maybe it does not matter?
No, I think it does. I think it probably does matter. It might depend on what the fragment is. And again, I kind of would like to have some biochemical collaborators who've maybe had a chance to think a little bit more deeply about it than my naive physicist self.
Okay.
But yeah, I think it probably does.
But do you think you have the statistical power? Like, do you have enough data to resolve this?
It depends on what data set we're working on. Possibly not actually in the antibiotics data. It's about 35,000 molecules, like I said, and we don't have a lot of experimental data. But possibly if we went and tested on a different data set, because there are some really huge ones out there with pretty huge experimental banks, yeah.
I should not monopolize the question, so is there any other question?
I have a follow-up question on that. So you mentioned when you were talking about your fragments that eventually, maybe down the road, what you'd like to do is say, okay, if you have this fragment on your protein, you can permeate it. And if you have this other fragment, well then it's going to kill the bacteria. Yeah. But it's unclear to me, if you want to combine those two together, if it's still going to work. So do you have any insight?
So it's an interesting question. I think you're right. Sometimes they combine in really weird ways, right? You can absolutely have nonlinear effects in there. What has been seen, interestingly, is that a lot of times if you have like two active things and you stick them together, they work better. Oddly, but it is possible that they wouldn't. And so essentially what I'm doing right here, is like a really linear approximation. We haven't considered second order or anything. And so that's something that I want to think about. I don't really have a good answer right now, but I think it is an important question.
Otherwise, I have a question on the toxic snails.
Yes.
So what happens in the actual venom, like, because you just showed us that there are like many conformations and like it's complicated basically, but what happens for the actual venom, like are the synthetic, I mean, is it the sequencing or is it the way the sequence of venom that makes it efficient?
It's folded in the endoplasmic reticulum.
Okay, yeah.
So there's probably a bunch of chaperones and things happening.
So
there's two things, right? So in vitro, we're not really mimicking you're not really mimicking the cellular environment. The other thing is, though, I think I did see some evidence. I think this was spider venom, but don't quote me on it. That they do have metastable ensembles in the animal, which would kind of make sense to me because then you kind of have somewhat more discriminative power, right? If you have multiple different conformations, you have potentially multiple different things you can bind to. And in fact, maybe that's why some of them are more one way and some of them are more another, just evolutionarily. If one of them is really impressive and binds to, I think, the sodium channel, blocks your sodium channel real good in the one conformation, on the others maybe it slides more towards ordered, but maybe there are reasons you would want, quote unquote, a metastable ensemble because you have you can be more general in some sense, right? But I don't know for sure because it's a very complicated little infrastructure in there.
Is there any other question? Okay. Don't be shy, guys. As always. I already asked 2 questions, so I don't want to ask too many questions.
I'm fine with no more questions. I'm very tired.
Okay, so I think then it's a good time to thank our speaker today.
I'm going to give one little shout out, sorry. I'm giving a talk on mental health and physics, a virtual kind of fireside anonymous questions, perfectly fine talk, virtually here at McGill-ish, virtually, on October 18th at 5 or 5:30 p.m. Did we ever decide? We haven't decided yet. At some point after the day. So if that's something you're interested in, it's happening.
Thank you. Thanks, everybody.